Next topic is DNA replication. You have already studied in depth microbiology and the cell division of microorganisms, so we'll not go in more detail about prokaryotes. Eukaryotes, anyway, you have seen it, but still we'll add something because we are more relative. It doesn't mean that you should not go for prokaryotes, but yes, you have studied it well, and it is expected that you are reproducing it, you are trying it, so that we'll able to concentrate more on eukaryote one. So you know what is DNA replication, and it is a very important stage in the cell division. So basically, what happens is very roughly when we say that two strands of the DNAs are separated. Now, when you have seen about this separation of DNA strands, then definitely you'll able to understand, if you remember, that how long the DNA is there. Last time we have seen it, that how long the DNA is, and this DNA is how it is coiled, how it is put, that is wound up on the various histones, and how the nucleosomes are there. Now, if we imagine this, or we can understand that, we have seen that a very big one structure has been put into a very small one, or we have seen that some centimeter length strand has been put into micrometer or even smaller than that cells and the nucleus. That means it has to be nicely packed. We have seen that packaging, and this packaging is very important. Again, that this packaging, if it is not proper one, if it is very haphazard, then what will happen is, when it has to be processed, then at that time this process will not be possible, and therefore we have got various activities. We are having one figure that's showing so many things which are there. All these things, again, the details we shall have when we are going for genetic. But then briefly, we have got two strands. These two strands are separated, okay, and one will call as a parental strand, another will call as a lagging parental strand. Normally when your DNA is there, we call it as a 5' 3'. One end will be 5', another end will be 3', okay, and same thing is there, the reciprocal one, the or we call it as a complementary one. So whatsoever complementary will be there, suppose we have got parental strand from 3', then another will be 5' to that, okay. So this 5', 3', they will be separated because of the various enzymes which are there. The enzymes are like DNA polymerases, like delta DNA polymerases, alpha DNA polymerases, primerases. Then apart from that, the topoisomerases, which are causing unwinding or of this. So the long strands are there, they are unwind. Because of unwinded, then the helicase are there, which will be the helical structure will be there, which will be separated. Then DNA stranding binding proteins are there, which are separated. DNA polymerases are there, okay. This enzyme is responsible for separation of the strand, and accordingly the primers will be made. RNA DNA primers will be made, and accordingly it will be there. This is very simplistic view we had so far. Now this view, when we see, we'll see what the details are. So it is the action of DNA polymerases. They are synthesizing a new DNA strand which is complementary to the original template, and this synthesizing DNA, the double stranded DNA, it has to be unwound by DNA helicases and because of the polymerases. Before that, there are topoisomerases, as we have seen. So they will be doing this topoisomerases, helicases. They will do it. They will make it at a straightening of the strand. Then what I told you, that is the wounding, whatsoever winding is there, that has to be separated, okay. And they will form the replication fork, which is containing, fork means a fork like structure, okay. where this just like comb like structure I can say. So this type of structure will be there. This structure will cause replication process. It will permit the copying of single DNA helix into two DNA helices, and they are divided into daughter cell at the time of mitosis process. Again we shall be seeing later after DNA ring. So this process involves so many enzymes. Okay, in that so many enzymes are there, and these enzymes which are carried out at the replication fork are well conserved from the prokaryotes to eukaryotes, and the replication mechanism machinery in the eukaryote DNA is much larger complex. It coordinating many proteins at the site of replications, and by that it forms the replisome. So what are the replisome? They are responsible for copying the entire genomic DNA into each proliferative cell. Now in proliferation again, if you remember last year in pathophysiology you might have studied, proliferation happens, and basically there are two important functions it is happening. One is cell division is there, and along with cell division cell differentiation is there, is one kind of proliferation. And another kind of proliferation is increase in the growth of cell. If the cell size was lesser earlier, then there is an increase in the cell size. So there are two ways by which proliferation takes place, and these are particularly useful for many physiological activities, including repair and other mechanisms. So this is happening in each kind of proliferative cells. Those which are having the proliferation capabilities, this cell will do all this activity, and this cause the passage of the genetic information passage from parental cell to daughter cell, and thus the new cells will carry all this information which is important for their function. Much of the cell cycle is built around the ensuring that DNA replication occurs without any error, because that is very very important. Anyway, we are going for more details about cell cycle and the phases. I will not repeat, I will not tell anything about this now, that is G1 phase and S phase or G2 phase, etc. But yes, these are various phases, they are taking place in the cell cycle, and they are playing very very important role. Okay, what happens in each cycle? After this replication we have got that topic which we will be discussing. So what happens in that? Okay, so there is a initiation of replication is the first stage of this. The DNA helix is unwound, and the initial priming event by DNA polymerase alpha, they will doing the leading strand. The priming event, that is primed there, and because of this priming on the lagging strand, that established the replication fork. This priming of the DNA helix that consists of synthesis of an RNA primer. It allows DNA synthesis by DNA polymerase alpha, and this occurs once at the origin of the leading strand. At the start of each Okazaki fragments are there. Now Okazaki fragments are the fragments which are not having I'll repeat the figure here. So these are the Okazaki fragments which are not having very important information, but they are playing important role, or they are part of the DNA, and they are intermittently present. So like that, parental strand and this lagging parental strands are there. This is replication complex is there, where alpha, beta, alpha, delta are there, primase is there, and after it is there, when there is a separation is there. So this is forming the a comb like structure, or as I told you, this makes the primers are available, and with these primers, these RNA primers are attached, and they are going for the leading the strands. So this is how we have seen this figure that explains briefly about the processes which are taking place, and these processes, they are going for the step by step, going for the formation of new DNA. It is initiated from the specific sequences, they are called as origin of replication. So we have got specific codons are available. You know codons. So specific codons are available. They will, when they are activated, they will cause the replication. And they have multiple replication origins, particularly in eukaryotic cells. So multiple replicative proteins, they are assembling on the that area, and they will dissociate from these replicative origins. The individual factors that are described here, they will be working together to direct the formation of the pre-replication complex, that is called as a pre-RC, and this key intermediate in the replication initiation process. The association of the origin recognition complex, or ORC, with the replication origin circuits, they will recruit the cell division cycle 6, or they are called as a CDC6 proteins, and they will form the platform for the loading of the mini chromosome maintenance MCM complex proteins. This will be facilitated by chromatin licensing and the DNA replicating factor 1 protein, that is CD1 type of protein. So ORC, CDC6 and CDT1, they will together, they are required for the stable association with the MCM2 to 7 complex, and they will make the replicative origins during the G1 phase of the cell cycle. So during G1 phase, what is happening, which we have described briefly at that time, again will not repeat it. This has happened during G1, but whatsoever other things related to G1 we can see again. Then this pre-replicative complex is there, again, which is happening during G1 phase, that they are making the pre-replication complex, pre-RCs, at the origin of replication. This involves the order assembly of many replication factors, including the origin recognition, that is ORC, CDC, CDT1 protein, and MCM227 protein. These all are assembled. After their assemblies, there are two important things are activated. Particularly you have to remember this, because this will be coming again and again at many places. So this causes by two important enzymes. There are kinases enzymes. One of these is called as a CDK, that is cyclin dependent kinase 2, and DDK, that is DBF4 dependent kinase. These important kinases are responsible for activation of the complex, which we have seen as a pre-replicative complex. They are very, very important. These two enzymes are there. And when we are going for next topic, after cell cycle, there is one more topic we have got. This topic is related to various pathways. Okay. Now, let me clarify over here is, the other pathways I will not go for. Our other important transduction mechanism will not discuss, because you have studied most of these in earlier classes. We shall be only concentrating more related to cell related to one. Otherwise, whatsoever receptor mechanism and transporters, it is there. It is already, you have studied it in detail. So, example, G protein coupled receptors and other, but only this kinase type of receptor, or it is also called as insulin type of receptor. We have got many signal transduction mechanism or pathways. So these pathways we shall be studying, because you have not studied them in details. But you should be ready with the earlier one, the other one, that is G protein coupled receptors plus maybe membrane bound ion channel receptors or nuclear receptors. Nuclear receptor again we shall be needing, because they are playing important role in this also. So this receptor mechanism you have studied earlier. You please just go through before we complete this topic, so that you will be able to understand how the nuclear receptors are acting. And on that basis only you have seen the mechanism of glucocorticoid mechanism and other steroid mechanism, because these steroids are acting by that mechanism only. So this is how we should try to link the things which we have studied earlier and make the things in a very synchronized manner. So these are the two important kinases, that is CDKs or DDKs, and these kinases, they are important for that. That involves the order assembly that assembly order, a very important word is there, ordered assembly. That means that assembly should be ordered. Uh, okay, if, suppose you must be knowing, or if those who want, many times WhatsApp videos are also there, that assembly of car making, or assembly of motorcycle making. So that means these assemblies are made in such a way that it is very clearly one after another parts are assembled into it, and that is ordered. So suppose if it is a car or motorcycle, then in first assembly the chassis will bring in, then somebody will fit something onto it, then next one will fit engine on it, next one will fit some the body part on it, or maybe fuel tank on it, the next one will be attached, make the connection of it. So this is what we call it as a ordered assembly. Okay, ordered means what sequence should be next to next will be there, and accordingly each, one after another, that makes the production faster and accurate. That is correct one. If it is not ordered one, then either it can affect it, or quality wise, or quality means it may not be the correct thing, or it may not be amount wise. So quality or quantity wise it can make that. So ordered assembly is very important. So additional replication factors will be there. It will be required for unwind the DNA and accumulate the multiple eukaryote DNAs into that. Just a minute, let me answer the phone. Hello, हाँ बोला मैडम. ओके, हाँ बरोबर. हाँ बरं बरं बरं, चालते चालते. ओके, हां मग आपल्याला मीटिंग घ्यायची गरज नाही. ओके चालतं. ओके, थँक्यू. Actually, additional responsibility is also there, having charge with me, principal charge. So anybody may be having some office related activity, I should answer them properly, timely. Therefore this is happening, and therefore I could not even conduct some classes in the meanwhile. I got myself free in the meanwhile. So this is why I have to answer the calls in between, because they are also having important work which may be hampered because of if I do not answer them timely. I am sorry for that, but this is the reason. Now this is how we have seen. So this orderly assembly is there. Then these DNA polymerases, they will be doing the unwinding of the DNA, and then one by another activities will be there. All these complexes will be playing role. Then next complex will be the origin recognition complex, that is ORC. This will recognize the origin of that. Okay, and this will be causing the next activity, that is in the late mitosis, it will be playing the role. And all again proteins what we have seen, like ORC, CDT6, CDC6, CDT1, they are all required to load the 6 protein which are responsible for MCM 2 to 7. See, this 2 to 7 means there are 6 proteins onto DNA, and this 6 subunit protein complex that will be selecting the replicative origin sites, and that will make the ORC binding to chromatin through the cell cycle, and accordingly this will be making impact, or this will be causing the next stages of that. So these are what are the next stages. These are studied actually from the microorganism or yeast, which is known to bind with this replicating sequences. So these yeast and bacterial processes are helping to coordinate, because they are easy. It is not easy for going for human being in more detail. So from this all details we get some information. So this information, what it gives, that when the ORC bind to the DNA at replication original, it serves as a scaffold for the assembly. Okay, that is a complex scaffold means, and this will assembly of other key initiation factors, and this pre-replicative complex assembly during G1 stage of the cell is required prior to the activation of DNA replication, and this will thus make the, just a minute, हाँ ठोंबरेजी, तुमचं अभिनंदन बाबा आणि तुम्हाला धन्यवाद.